Good afternoon, Clement. How are you today?
Good. How about you, Richard?
Doing well, thank you, and thanks for joining us. Folks, we're here with Clement Chow, and welcome, Clement, to Rare Awareness Radio. And thanks to Jill Hawkins from the FAM177A1 Foundation, Research Fund, and Foundation for connecting us. I'm excited to speak with you, and it's been a little long. It's been a little bit of a long time coming, hasn't it? But we finally able to get together, which is the most important part, right?
Yep.
Yeah.
Excited to talk with you today.
Absolutely. Maybe we could just start with an intro. You could tell us a little bit about yourself, and where you're currently located, and a little bit about your background. It's always great to orient folks to that.
Sure. Yeah. So my name is Clement Chow. I'm an associate professor in human genetics at the University of Utah School of Medicine. I've been here about almost 10 years now, and my lab is really focused on trying to find new therapies for rare diseases.
Hmm.
We use a number of different models, mostly the fruit fly, Drosophila, and we have this real passion to try to help the rare disease community.
I love it, and I am so thankful for men and women like yourself who are leaning in so heavily to the rare disease community with your research. So you mentioned the fly model, and the work that you have done around FAM177A1 and the fly model. Would you walk us through a little bit about the deficient fly model? How did you kind of come up with this? Challenges you've been facing? I'm fascinated by it.
Yeah, so our my lab takes the approach that this fruit fly, Drosophila, kind of this pest that we have, on our countertops eating our bananas, is actually we think is a key to really helping a lot of rare diseases.
Hmm.
The f- the fruit fly has been kind of this I think unsung hero of biomedical research for a while. Lots of labs work on it. It's a great tool because it's fast,
Hmm.
it's cheap to use, and we can generate so many of them in such a small space. Very very different from mice, for example.
Mm.
And for all those reasons, it's a really great tool for biomedical research, but it hasn't really been used for drug discovery
Mm.
very much until more recently. And my lab has, in the last few years, has really kind of run with this idea that we can use this fruit fly, this kind of humble insect, um, and use it to try to find new drugs for people living with rare diseases.
Mm.
And so when Jill came to us and said she'd like to work with us to try to do this exact thing for FAM 177A1, we were thrilled to kind of work with her and help her out. Up to up till then, we had done several different kind of drug screens using the fly model, and it worked out quite well. So we were kind of excited to jump right in. And so the people living with FAM177A1 mutations are what we call loss of function.
Mm.
They have reduced function of this gene, and so we set out to generate a model of this disorder with reduced function of the same gene in flies. The really nice thing about the FAM177A1 gene is that it's conserved in all animals, and so it's really easy to kind of generate a model, a one-to-one
Mm.
model of the human disease in the fly. And so we
Mm.
did that. We use kind of pretty standard genetic tools in the fly. Another kind of really nice thing about Drosophila and fruit fly is that there have been a ton of tools that have been generated by the community that are readily available to any lab using them, and we like to take advantage of that because anything that reduces cost of these screens is really a plus in the end for these rare disease families. And so we pulled a bunch of kind of standard genetic tools that we can use in the fly and generated a loss of function model of FAM 177A1. And as you can imagine, it's not good for the fly to lose function of this gene. Just like it causes a neurodevelopmental disorder in humans, it causes something fairly similar in flies. And when we look for when we do drug screens, we want to look for what we call phenotypes or things that go wrong in the fly when you lose
Mhm.
that gene. Lots of things can go wrong when you lose any gene. When you lose FAM177A1, lots of things go wrong, but not everything is very useful for a drug screen. So we have a list of what we call phenotypes that we like to use for drug screens, and we just basically run that model that we created through that gauntlet of phenotyping and just find the best possible phenotype that might work for a drug screen. And in this case, it was actually what we like to call lethality, which is just death.
Hmm.
And in these flies, when you lose FAM177A1, the flies make it through development and make it almost to adulthood, and then they die.
Wow.
And dying sounds really terrible,
Mhm.
especially in the rare disease
Right.
context. But in this case, actually, dying is a great phenotype for drug screening because you're really forcing a drug to rescue the death of an organism, as kind of as kind of the positive outcome.
Sure.
And so we developed this model. We had this kind of lethality phenotype where the flies die right before they become adults. And we basically simply asked, can a drug make these flies live past that point?
Hmm.
Right. It's a very distinct point in the development of the fly. So it's very easy to identify whether flies live past that point or not. And then we just throw all the drugs at them. We did about 1,500 drugs.
Oh my gosh. Wow.
FDA approved drugs. In this, what we call repurposing, which is the use of these drugs that have already had previous uses identified, and this of course moves things to the clinic a little faster. But we threw 1,500 drugs at them and found a number of drugs that actually allowed these flies to live past that point that they
Wow.
normally. Yeah, and this is a lot of work. I have a really talented group of. Yeah, it's a lot of work. I don't personally do it, Richard.
Right.
I have a talented group of scientists who run these drug screens, whose job is just to run drug screens for rare disease families and foundations, and they did all the hard work for this. So shout out to them for sure. But
I,
we
I,
ran.
I'm
Go
just
ahead.
trying to wrap my head around how you worked through how your team conducted a drug screen of 1,500 drugs. I'm still stuck on that point, Clement.
Yeah, no, let's talk about it for a second.
Yeah.
So, we've done this for a couple of years now. So we've actually really streamlined this process, and we have a couple technicians, and the food is we put flies have to live on this food, right? And so we put
Right.
their food in tiny vials, and each of the vials is spiked with one of the 1,500 drugs, and the flies lay their eggs on there, and they grow up, and then we simply see if they live past that point that
Right.
we know that they die, right? And because it's a lethality kind of survival type assay, it's really easy to know whether you
Hmm.
see something living or not at
Hmm.
the end. So it's actually, it's a lot of work for sure, but it's also not as much work as some of our other screens. We're just looking for presence or absence, right? And so yeah, that's how the screen is done. And we ran that screen. We found a number of drugs that seem to really kind of have some benefit in the survival
Mhm.
of the FAM177A1 flies that we generated.
Outstanding. Outstanding. You mentioned the lethality phenotype, but I know from when you and I spoke previously, some of the more near-term applications and near-term impact you were looking at as well was symptom management. Um,
Mhm.
how does what you're doing, how do the findings translate into some of the symptom management? I'm curious about, Clement.
Sure. That's a really good question. That actually is a question we get a lot because people it's hard for people to kind of visualize whatever we're doing in a fly has anything to do with a human,
With a
right?
human being, yeah,
And
yeah.
yeah, right. And so we generate this model in the fly, and of course a fly is not a human, but the core process of what that gene FAM177A1 is doing is the same in the fly and the human, right? The cell
Mhm.
biology is the same. So even though the organismal output is different,
Mhm.
the gene function is the same. And so our drug screens are looking for the rescue of that defect that ultimately gives us lethality in flies or a neurodevelopmental disorder
Got
in humans. And so, what we're measuring in flies might have nothing to do with kind of what we see in humans, but it still fixes the core problem
Okay.
of missing that gene. And
Mm.
so, when you mention symptom management, there's a lot of things a drug can do, right? It can replace the loss of that gene. That's harder
Mhm.
to do.
Mhm.
But what's more common is that a drug can reduce symptoms, right? It
Mhm.
can reduce some downstream or secondary effects of losing that gene, many of which might be positive for a person living with this particular disorder or any rare disease for that matter. And
Mm.
so, being able to reduce things like, just as examples, reduce things like seizures,
Right.
or improve, cognitive function, or improve kind of ambulation or walking. All those things are positive, but they're also just symptom management and may or may not be actually addressing the core issue.
Yeah.
But when we think about therapies for rare diseases, it's an all, everything is in the cards, right? Like
Right.
we need things that manage symptoms. We need to move towards things that cure the disease, and we just need every tool we can get
Yeah.
our hands on.
You
And
can
so,
throw at it. Yeah.
yeah. And
That's
so going into a screen, we don't know what we're gonna find, but,
right.
I think symptom management is a pretty good goal. I think, yeah.
And you mentioned the underlying, right, and that's kind of one of the things I also wanted to ask you about, Clement, is as these, you go beyond identifying system targeting therapies or symptom targeting therapies, and you start to get into the, underlying disease. How much closer are you to understanding the FAM177A1 deficiency and its evolution towards addressing the underlying disease?
Yeah, this is a great question. So one of the biggest challenges of rare diseases in general is that they're so understudied, right?
Hmm.
Many of these genes we don't know much about, and
Right.
that's certainly the case for the gene FAM177A1. We actually don't know very much about what it does. We kind of have an idea of where it might be and what it's kind of doing to the cell, but we don't really have a good idea of
Yeah.
of what it's doing kind of on a day-to-day basis. And so that's really tricky, right?
Right.
Without that knowledge, you find a bunch of molecules that seem to improve the function. You don't really know kind of how that intersection is happening. But what's really nice about these kind of screens is that we don't have to know anything to find something positive,
Hmm.
right? It's based on the fact that there's something broken, and we would just want to find something that can fix it. And the fact that, yeah, right, like it's really simple
Yeah.
actually. And
Yeah. It makes sense.
the fact that this particular molecule or that particular molecule or this class of drugs can improve the outcome
Hmm.
tells us that whatever that drug is doing probably is likely very important to the pathway that
Yeah.
FAM177A1 is involved in, right?
Hmm.
And so then you can start marching backwards in more of kind of like a standard basic science research project, and kind of really work out what that mechanism is, and maybe even get to a point where this drug tells you exactly what happens when you lose
Right.
FAM177A1,
Right.
right?
Right. Exciting stuff. Clement, is there a natural progression here or a logical progression here? You mentioned at the beginning of our conversation how using fruit flies is highly effective. It's highly cost effective. Um, but is there a transition here into mouse models or cell cultures or other systematic ways to validate your findings that you have kind of started to identify, or is that kind of something someone else will focus on? I'm curious as to how you see the progression to other models.
Yeah, so I think that all those are possibilities.
Hmm.
I think that we undertook I mentioned this earlier, what we call the drug repurposing screen, right? The
Yeah.
the use of FDA-approved drugs, and kind of the beauty of that approach is that these have all safety profiles, and they've been approved for some kind of use in human beings. And so the kind of the big dream of drug
Hmm.
repurposing is that you skip many of those steps that
Right.
take years and millions of dollars of funding to
Right.
get through. And so the hope is that when we find a hit in a screen like this, we can actually move very quickly into the clinic,
Hmm.
right? Whether it's kind of an N of one type study, or a bigger study, it allows kind of us to move in that direction if something makes sense and it's safe, etc. But certainly, it makes, we've done screens in the past where we didn't feel like we knew what was going on with the hits, and so it made sense, for example, to go back to some human cell culture models, or maybe
Hmm.
even the mouse.
Hmm.
Um, you know, all those are options, and I think we leave them all on the table.
Sure.
And sometimes it means that my lab does it, and sometimes it means that we
Someone
collaborate
else.
with
Yep.
with another lab. But certainly all that is on the table. But the real goal of this again is to really kind of move as fast as possible
I
through
get it. Yeah.
through to the clinic.
Yeah, I get it for sure. And I guess, and speaking of moving fast, the FAM177A1 Research Fund played a significant role in this project, and you talked about that collaboration with foundations and how important that is. As you scale, it seems like there is tremendous potential in the work that you're doing, and you are trying to go as fast as you possibly can, as you mentioned, and I'm sure you're trying to think of ways to accelerate your research even further and innovative approaches or technologies that you're applying to kind of supercharge these efforts. But what are the needs there, I guess? I mean, I think that the obvious need is probably funding, right? But what else? Where does it go beyond that, Clement?
Yeah. Yeah. That's also a good question. We've worked quite hard to streamline this kind of using kind of human might, right? And kind of
Yeah.
just willpower in my lab. And that's worked out quite well. And we actually can move at a pretty good pace,
Mhm.
but we want to be faster, right? And we want to
Right.
be more nimble. And so, we've been floating ideas about, how do we improve the throughput or the high throughput quality of our phenotyping. For example, a lot of that still is hands-on.
Mhm.
Um, the fly community, biomedical community, hasn't really developed many high throughput screening techniques like you have in cells. For example, you can
Sure.
imagine it's a bit harder in a 3D fly than it is in a flat cell, for example, right?
Mhm.
And
Mhm.
so there's some technical challenges to that, but we certainly are thinking about how can we improve that 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 speed on that side of things.
Yeah.
And and I think that some other things we've been thinking about really is questions we've been kind of asking ourselves is, do we need to run a screen for every disease?
Right.
What if FAM177A1 is in a pathway of 10 other genes, hypothetically, right?
Mhm.
Maybe things that work for FAM would work for those other disorders.
Yeah, sure.
We've
Yeah.
been Yes, yeah. So we've been thinking about, can we take the disorders that we've already screened for and take the top hits and just apply them to the other disorders in those pathways and ask
Yeah.
if they work, right?
Mhm. Yeah.
And so this kind of like clumping and making things a little less rare, right?
Mm.
In terms of thinking about therapies, I think is probably a next step. And obviously we're not the only one thinking about this, but it is kind of probably the next wave of how we think about rare diseases for sure.
Yeah, again, I did want to ask you, as you thought about your research contributing to the broader, right, rare disease discovery field, or the broader implications beyond FAM 177A1. Sounds like you're already sort of percolating through some of that today.
Yeah, for sure. Yeah, we are. And FAM177A1, we completed that more than a year ago now.
Mm,
And we've done more than 20, nearly 25 screens now. And there are definitely patterns that are kind of showing up, and we're thinking about how to apply those patterns.
right.
Right. And I should also mention kind of the broader applicability of some of the work we're doing is that obviously we want to find a drug that can move to the clinic ASAP, right? But even in the situation where we don't find a drug that's obvious to give to kids in the clinic,
Mhm.
that drug can blow open the biology for that disorder, right?
Mhm.
It provides, we go from nothing to a list of drugs that improve a phenotype in a model organism. A lab can take that and run with it and really blow open the biology of that disorder.
Mm.
And so these screens are not only good for therapies, but actually just good for learning about these diseases, right? And that can
Right.
have a pretty big, long-lasting impact on a
Mm.
particular field as well.
Mm. How do we get more folks sort of excited in the kind of work that you're doing, as you think of the next generation of geneticists, genetic medicine, genetic scientists, to kind of think more about focusing on underdiagnosed diseases, underrepresented diseases, rare diseases? Is there something that maybe just theories on how you attract people to your work in your labs? I'm
Yeah,
curious about that, Clement.
yeah. I mean, I'll tell you what, I think the next generation of scientists, the current students, are really excited about this kind of work. They want to
Mm.
make an impact. They
Yeah.
want to meet they want to meet the patients behind these projects that we have, right?
Right.
And I think that the more, you know obviously basic science is incredibly important, and we
Right.
learn a lot, and we make lots of important discoveries with basic science. But the more we can kind of keep in mind that there are people behind these disorders, right? There are families behind these
Yeah.
disorders,
Yeah.
that I think that can really drive the work, in a way that publishing or even getting the next grant really doesn't do it, right?
Mhm.
And I think that what we're doing isn't earth shattering, right? We're not doing something that requires lots of fancy equipment or whatever, but it's a way of thinking that hasn't at least hasn't been a very popular way of thinking about science, at least in the fruit fly community, and in other communities. And a lot of this has to do with how we think about funding science, right?
Yeah.
Yeah. And how scientists are forced to spin projects a certain way or ask questions a certain way to get
To
funding,
get
or
funding.
even
Yeah.
get things, yeah, or even to get things published, right?
Right.
And so there's a lot of that is systematic, right? Of course,
Hmm.
but but I think but I think change takes time, but kind of remembering, I think we're really remembering why we're doing this work, I think can help at least push the next generation into being more creative, and how we partner with families, how we partner with foundations, and kind of get
Hmm.
this work done.
I couldn't agree more with what you just said about, this next generation of scientists. They want to meet the patients, right? They want to see that impact, to your point. And a lot of the researchers like yourself and scientists that are working so hard that we've certainly talked to in our small N, have a very similar refrain there, right? It's really that opportunity to make significant impact and know the patients and meet the patients that you're working with is really driving them, so it's exciting, and it's very encouraging to see, because you think, well, I want to work on a big thing, I want to cure cancer, right? I don't, they're not necessarily thinking about a rare disease, so it's great to see that
Yeah.
that's happening. Where are you, what's next for you, I guess I would ask, Clement, as you continue your work, what's kind of on the immediate horizon, things you're excited about, and what's next up on the docket, I guess.
Yeah, yeah. Yeah, so we're continuing to do these screens, and we keep getting interest
Yeah.
from foundations, and I have a couple emails I have to answer in my inbox about this, right? And so there's still a lot of opportunity, and we're still just scratching the surface of what we can find. We certainly haven't exhausted this
Yeah.
approach at
Right.
all, but really moving forward, we really are thinking about ways in which we can kind of cut down the work, right? Like, how
Hmm.
much can we lump? How much can we make things less rare, right? And so that's really kind of, I think, the next move for us.
Hmm.
And just to kind of give a plug for the other side of my lab, we've been the other side of my lab, which is kind of the bread and butter of what we do, kind of the more basic science, is we think
Yeah.
about modifier genes and how these genes are impacting the outcome of a particular rare disease, right? And so for example, all these people with FAM177A1, they are all actually a little different from each other, and some of that is environment, but a lot of that has to do with what else is floating around in your genome.
Mm-hmm.
And we've been studying, the effects of these so-called modifier genes in the genome, and how they impact rare diseases, and what these modifier genes are is they're drug targets. And
Yeah.
we're also moving towards kind of flipping some of this genetic knowledge we have into drug targets. And what we're finding actually is that in the cases where we've done both one of these modifier screen studies with a drug study, that they tend to, the Venn diagram intersects. We
Mm.
find drugs that target the modifier genes that we find in these seemingly unrelated studies. And so it really tells us that kind of this two-pronged approach to try to find drugs, one that's kind of obviously looking for drugs, and one's kind of going through a different direction to find genes first, can really get us there, and really could give us a lot of power in improving therapies.
I appreciate you giving us that little insight, like you say, into the other side of the coin and the basic science that your lab is doing complementary to the Drosophila and what you're doing around organism research. So thank you for that. I think it's very, very enlightening, very insightful. We've been talking with Clement Chow all the way from Utah. Thank you so much for giving us a little insight into the work that you are doing. And thanks again to Jill and the FAM177A1 Research Fund and Foundation for making this connection. We really appreciate you taking some time out of, I know, what is a very busy schedule, to share some of your work and your insights with us, Clement. So once again, thank you for your time and best of luck. And we'll be continuing to watch what you folks are doing.
Thanks, Richard. This was really fun.
Awesome.
Appreciate being on.
Thank you.